Hello everyone, my name is Lakshmi, PhD student from the University of Mauritius. I am going to talk about the use of machine learning algorithms to predict cell-material interaction in nanoscaffolds for skin tissue engineering. Nanoscaffolds are used as support to promote wound healing and skin regeneration, just like platforms that support workers in building construction. The latest generation of nanoscaffolds have the ability to respond to stimuli and encapsulate molecules. The challenge today is to match scaffold materials and scaffold design because when implanted in the body, a scaffold encounters multitude of cells and molecules. The performance of a scaffold is characterized by its physico-chemical parameters and its biological property, and our experimental work showed no linear relationship between these characteristics. So a preliminary study using machine learning can help us to better understand cell-material interaction and facilitate this long research process. The goal is to predict the biological outcome given the physico-chemical properties. For this study, we used data from 13 families of scaffolds. We built a simple predictive model and trained 6 supervised regression algorithms. The physico-chemical parameters, pore diameter, fiber diameter, contact angle, and Young modulus were used as features for our ML model, and the biological property, which is the number of cells grown on the scaffold during in vitro experiments, were used as target variable. So 80% of the data were used for training and the remaining 20% for testing. We tested 6 algorithms and performed hyperparameter tuning to get the optimal parameter values of each model and the best performance. Let's now have a look at the results. So after hyperparameter tuning, random forest regression was the model with the highest accuracy of 62.7%. The correlation between actual and predicted output data showed a significant descent fit, which means a potential relationship between the predictors and the outcome that can of course be improved with more data. Another interesting result from feature selection analysis indicated that fiber diameter and pore diameter impacted more on the target variable. Outputs from the ML model were also correlated with data from in vivo experiments conducted on rats, and we noticed that scaffolds with small fiber diameters enhanced the formation of blood vessels during the wound healing process. So this is the first study from our research group on machine learning methods to predict scaffold performance for skin regeneration. Future work would be testing other algorithms to improve the accuracy of our model. We are aiming at a final generalizable model that will take scaffold parameters as input and predict the number of skin cells in novel scaffolds. In parallel, we are also following the wound healing process to study other types of cells. Thank you for your attention.